Hi everyone. We'll be studying restriction enzymes today. This is a part of your chapter 11 for class 12 students, and it's a very important tool for recombinant DNA technology. Now, you're already aware about genetic engineering, gene manipulation, and you know the structure of DNA from your previous unit. Going ahead, genetic engineering is a lot about manipulating DNA, which means you have a DNA strand, you cut that DNA, take that small piece of DNA and you insert it into another DNA strand. So you are manipulating the original DNA strand, inserting and making an entirely new sequence of DNA. But DNA is not something that you can hold in your hands or even see for that matter. So how are you going to manipulate? You're going to need a very special set of tools which are known as restriction enzymes. Now, these restriction enzymes are very appropriately and commonly also known as molecular scissors or chemical knife because they are being used to cut DNA strand. DNA is a molecule, so they are known as molecular scissors or chemical knife. So our learning goal for today is understanding what are restriction enzymes. A restriction enzyme cuts the DNA double helix at specific sequences. So these are site-specific cuts that are being made by restriction enzyme. Let's see how. So our learning goals are discovery, nomenclature, mode of action, and uses of restriction enzymes. Now, let's begin with discovery. Restriction enzymes are a part of restriction modification system which was found in bacteria. Bacteria uses this as a defense mechanism against foreign DNA. For example, if a bacteriophage infects bacteria, bacteriophage you know is a virus that infects bacteria. The restriction modification system degrades the bacteriophage DNA before it can damage the bacteria. So you can see a bacteriophage enters, injects its DNA inside the bacterial cell, and that DNA is immediately degraded by this restriction modification system. So as a part of this restriction modification system, Werner Arber in 1960s discovered restriction enzymes. They were awarded Nobel Prize for the discovery and isolation of restriction enzyme. The first restriction enzyme to be isolated and characterized was HIN2. What is this restriction modification system? This comprises of two enzymes working together, the restriction enzyme and the modification enzyme, which is the methylase. This restriction enzyme restricts the growth of bacteriophage. The bacteriophage which is infecting the bacteria is going to insert its DNA, and the restriction enzyme, as we just saw, degrades that DNA by cutting it at its phosphodiester bond. But how is this restriction enzyme going to differentiate between its own DNA and that of the virus? For that, it uses a modification system involving methylase. This methylase modifies its own DNA. That is the DNA of the bacteria. You can see here there is the bacteriophage DNA and the bacterial DNA. The methylase will add a methyl group to its own DNA, that is the bacterial DNA. Not just its own DNA, not anywhere. Methyl will be added at that same site where restriction enzyme recognizes the sequences. Those same sequences which are recognized by restriction enzyme will have a methyl added by this methylase. Now, as a result of this armor provided by methylase, the restriction enzyme will be able to identify that Methyl is present, so this is my or bacterial DNA. Methyl is absent, so this is the bacteriophage DNA. Now, because of this modification, bacterial DNA is protected from degradation by this restriction enzyme. Bacteriophage DNA, which does not has methyl group, is degraded by the restriction enzyme. Restriction endonucleases belong to a big group of enzymes which are known as endonucleases. Nuclease means an enzyme which cuts DNA. Endo means it will cut DNA anywhere along the length of the DNA, anywhere in between. In contrast, we also have exonucleases. Exonucleases will cut DNA only at the ends, nowhere in between. Now, these restriction enzymes recognize very short sequences of 4 to 8 base pairs, and these sequences are known as palindromic sequences. What are palindromes? We'll come to it later. So these 4 to 8 base pair sequences recognized by restriction enzyme are known as palindromic sequences. There are two options now. Whether the cut is between or within that 4 to 8 base pair sequences or somewhere close to it. So restriction enzymes are of different types based on whether they cut DNA within these 4 to 8 base pair sequences or somewhere close to it. Now, they cut in the presence of magnesium ions and are usually homodimers, which means they have two similar units so that they can cut the two strands of DNA, the double strand, simultaneously. Now, on the basis of features of restriction and modification, we are going to classify the restriction enzyme. Here I am taking only the restriction features. On the basis of the restriction features, there are different types of restriction enzyme. The first one, type 1. This cleaves DNA 1000 base pairs away from the sequences that it recognizes. So we have the 4 to 8 base pair palindromic sequences that are recognized, but it will not cut within those sequences. It will cut 1000 base pairs away. We have type 2, which cleaves at the recognized sequence within the palindrome. Type 3, that will cut DNA 24 to 26 base pairs away from the recognized sequence. And we have type 2S, which cleaves DNA 20 base pairs away from the recognized sequence. Now you know the most useful for us for genetic engineering is type 2. Why? Because we know where the cut will be made. That is very important for genetic engineering, to choose, to decide, and to know where we want the cut. If we don't know, if it is random, if it is 1000 base pairs away, then our purpose is not solved. So for genetic engineering, we are going to use the type 2 restriction enzymes. So restriction enzyme 2 or type 2 restriction enzyme is the most suitable for gene manipulation. Why? Because many of these make asymmetrical cuts. Why? We'll understand in a bit. The methylase and restriction functions involve two separate enzymes and they do not require cofactors. But most important, it cuts DNA within the recognition sequences. Now, in order to understand the mode of action of restriction enzymes, let's understand palindromes first. Now, palindromes are words that read the same from both ends. In case of DNA is double-stranded. The two sequences are complementary to each other. So, situation is a bit different. Here, the recognized sequences show twofold axis symmetry, or we say rotational symmetry. That is, they read same on the two complementary strands. You can see here in the top sequence GAATTC 5' to 3'. The second one, anti-parallel complementary strand GAATTC from 5' to 3'. These sequences are known as palindromic sequences. This GAATTC is recognized by EcoR1, the restriction enzyme of E. coli. The site at which this restriction enzyme cuts can be either this, then this type of cut is a symmetrical cut and it will produce two equal segments, GAA here, CTT here, base paired with each other. This will produce TTC here, AAG here, base paired with each other. So we have two complementary equal strands that are cut because of this symmetrical cut. The cut can also be like this, asymmetrical. Now, this asymmetrical cut will produce hanging sequences, and these sequences are more useful to us. Let's see why. Now, these are the sites of asymmetrical cuts by EcoR1 restriction enzyme. You can see that in both the strands, the cuts are between the same two bases, G and A. So, as a result, two fragments are formed. This leaves the TTAA here without any complementary bases to pair. Similarly, TTAA here without any complementary bases to pair. Now, these bases have a tendency to pair if they find complementary sequences. So, these become sticky ends, or we say cohesive ends. They are looking for complementary bases to form these hydrogen bonds to form base pairing. Now, when we are creating recombinant DNA, these sticky ends become very useful to us. If we have two different DNA strands, we cut them with the same restriction enzyme. They will both have the same sticky ends. And when we bring them together, they will automatically pair. It is highly probable that they will join to each other through complementary base pairing. That is why type 2 restriction enzyme, which make asymmetrical cuts, are most commonly used for genetic engineering because of these cohesive ends. Now you can see the mode of action that we discussed just now. This is your first DNA and this is the second one that needs to be inserted into the first one. You have cut them both with EcoR1. So this one also has unpaired TTRA. This one also has unpaired TTRA. So sticky ends of both are there. Now you can understand why we call them sticky end, because when we mix them together, it is highly likely that A of this will bond with T of this, T of this will bond with A of this, and we get the recombinant DNA because of this ligation. Nomenclature of restriction enzyme was given by Smith and Nathans. The naming is done to give information about the source. For example, in the restriction enzyme EcoR1, E comes from the name of genus Escherichia. Co comes from the specific epithet, that is coli. R comes from the name of strain of bacteria, and the Roman numeral 1, EcoR1. 1 is when many restriction enzymes are isolated from the same strain, they are denoted by Roman numerals on the basis of order of discovery. Remember, not isolation, discovery. So EcoR1 is the first restriction enzyme that has been discovered from E. coli bacteria. Now here are some common restriction endonucleases that are used in genetic engineering along with their sources. Okay, and now a few questions to check your understandings. How is the action of exonuclease different from that of endonuclease? Before seeing the answer, try to answer it yourself. Exonuclease cuts DNA sequences at the ends, and endonuclease can cut sequences anywhere throughout the length of DNA. One more, hint 3. What do H, in, D and the Roman number 3 stand for? H stands for Haemophilus, the genetic epithet. In, influenza, specific name. D is the strain, and 3 indicates the third restriction enzyme from the same strain of, that is the D strain of Haemophilus influenzae. Write specific palindromic sequence or palindrome recognized by EcoArban, GAA, TTC, and I have also shown you the site of the cut. Few multiple choice questions. The recombinant DNA technology has been made feasible with the discovery of restriction enzymes that are a part of defense modification system in bacteria. Identify the incorrect statement with respect to restriction enzyme. These are the options. Okay, the answer is C. Restriction enzymes identify palindromic sequences and always cuts within the sequence. It's not always. Sometimes it is thousands of base pairs away. Sometimes it is 20-25 base pairs away. So it is not always within the sequence. Now, these reason assertion questions are being asked now in CBSE exam also. So let's see one of these. These are the way in which we decide whether we choose A, B, C and D. Now question 1, assertion. Restriction enzyme cuts the strand of DNA to produce sticky ends. Stickiness of the ends facilitates the action of the enzyme DNA polymerase. So answer, assertion A is true, but the stickiness of end has nothing to do with DNA polymerase. So reason is false. So the correct choice will be C, assertion true, reason false. Another one, assertion EcoA1 cannot be used for genetic engineering. Reason, EcoA1 is a type 1 restriction enzyme. Now we just did this. Assertion A is true. The reason 2 is true. Type 1 restriction enzyme will cut the DNA strand 1000 base pairs away from the recognized sequence. So they are not of much use in genetic engineering. So the correct choice would be A. Now I hope you were able to understand a little bit more about restriction enzyme. In case of any doubts, any questions, drop a comment, get in touch with me. We'll try to solve the problems together. Thank you. Bye-bye.